welcome to this Allergy Research Showcase. We're trying to bring the latest from Imperial, and this is the third year we're doing this. And this year we have great colleagues presenting. So this is a little program for today. We're going to have Dr. Paul Turner giving about a 20-minute talk on the latest research from him and his group on food allergy. We're going to have about 5 minutes for a Q&A with Paul, and then we're going to have Professor Adnan Khan again for a 20-minute talk on disease trajectories in allergy, and again another 5 minutes Q&A. Many of you have probably heard Paul and Adnan, and probably that's why you're here, because we all love hearing from them, and we're very lucky to have such brilliant colleagues at Imperial. And this is part of the module leads we have in the program. So outstanding academics, outstanding clinicians, really working really, really hard and very, very committed to research and also to education. And I'm very privileged to work with them to deliver the allergy MSc. So I have a slide on the allergy MSc in case some people in the call might be interested. So this is the postgraduate education program we offer at Imperial. It's a part-time program, and in the first year you will complete a postgraduate certificate, and you see the different modules there, from the scientific basis of allergy to diagnosis and treatment of allergic disease, and then the cutting edge of allergy. So we really whiz through from the immunological mechanisms and the origins of allergic disease to the very clinical and practical elements, and then the latest research and research skills to develop your own projects. And we have an online stream now in addition to the blended stream. So if you wanted to do a fully online program, we can offer that now. In the second year, we offer specific modules covering a number of disease areas such as food hypersensitivity, rhinitis and immunotherapy, asthma, allergic skin disease, and pediatric allergy. And that's the postgraduate diploma together with the year one modules. And then lots of our students follow on doing a research project, often with us, with people like Adnan or Paul, and complete the MSc. And we have scholarships available. I'm sure my colleagues will put the links for that in the chat if there's interest. And we have an online open day for information on the 14th of June. Some people might want to join for that as well. So that's all I wanted to say as a little introduction on the sort of the education program at Imperial. And I'm going to hand over to Paul with a little introduction there as well. So again, many of you know Paul. Dr. Paul Turner is a reader in pediatric allergy and clinical immunology. He's a clinical scientist and an honorary consultant at Imperial. He's also clinical associate professor at the University of Sydney, Australia. And his research funded by a number of public bodies, such as the MRC or the European Commission, focuses on the pathophysiology of severe allergic reactions to foods. Paul also leads the food allergy desensitization program at St. Mary's Hospital, and he is a key member of a number of international and national organizations and initiatives in allergy. He has received a number of awards over the last few years, including the 2025 award from EAACI. So over to you, Paul. Looking forward to hear the latest from you.
Marta, thank you very much for that, as always, very kind introduction. So my brief was to tell you what I've been up to and the team's been up to over the past year or so. But I'm beginning to wonder in food allergy, really, if there's anything new under the sun. And for those who come from a biblical perspective, you might recognize that quote. I'm currently in Ireland, a hotbed of Catholicism, or certainly used to be. So it seems to be very debatable these days now. This is my conflicts of interest. There isn't anything relevant that's going to impact upon what I'm discussing today. What I am going to be doing is just quickly muting my email, because I can hear my NHS email busy pinging like mad, which is unhelpful for the purpose of giving this talk. Okay, all right. So if we, those of us who are familiar with food allergy and the direction things have gone in will know that the exciting thing apparently at the moment is immunotherapy, food allergy desensitization, or treatment for a food allergy. And if you just quickly pop into PubMed, immunotherapy, food allergy, you can see there's been a little bit of an explosion in terms of how many papers are now getting published on this. And these are original research articles rather than just reviews. But it's not really a new thing. You can go back to the Lancet in 1908, where Dr. Schofield reported the first known example of milk allergy desensitization in a 13-year-old boy with egg allergy. And subsequently, just a couple of years later, Dr. Schloss from New York likewise also published some information about treating people with food allergy. So certainly, it's not really new, although I think it's probably new in terms of everyone's excitement levels at the moment. And for those people who are biblically minded, this is a quote from Ecclesiastes, and sometimes I feel a little bit like this can be applied very much to food allergy, that there's really nothing new, that there's a thing of which one will say, see, this is new, but actually it's already been there long before we're here. There's no memory of it before. And likewise, in the future, people won't remember what we're doing now. And you can sort of think about, well, what's the point? And that's sort of what Ecclesiastes is sort of saying for part of it. So what have I got to contribute, and what's the team been contributing over the past couple of years? And I think one of the key things, and I think at Imperial, we're really fortunate to have some really dedicated members of the research team, not just myself, but all my colleagues, both medical nursing and admin supportive roles as well. And I think the key thing is that for us, our mantra almost is that we're not really smart, we just stay with problems longer. And so I'm going to be talking about how common food allergy is, correct diagnostics, immunotherapy, and severe allergy. And these are sort of four themes that we're working on at the moment. So in terms of the first one, epidemiology, some of you may be familiar with this. This was published around 2 years ago, and it was data from the NHS hospital admissions. And one of the problems we have in food allergy is it's very difficult to work out how common food allergy really is. To do that properly, because the testing is quite rubbish, 1 and 2 allergy tests which are positive, actually false positives, they're wrong. People have a positive antibody test for food allergy, but they're not actually allergic. And so as a result of that, we tend to use these surrogate measures like going to hospital because you've had a reaction or something like that. And the reasons for those is simply because we can't do food challenges in thousands and thousands and thousands and thousands of people. So we have to come up with the next best thing. And so typically people often go, well, more and more people are pitching up to hospital with anaphylaxis due to food, and more people are pitching up to hospital and being admitted to hospital because of food anaphylaxis. And you can see that in the chart on the left here. But I think what was really important and what was very reassuring with this data analysis is the fact that despite the fact you've got this apparent increase in the terms of people coming to hospital, maybe that represents anaphylaxis, maybe that just represents more people with anaphylaxis going to hospital and not staying at home, who knows? We are having a reduction in the fatality rate. So you're less likely to die from food anaphylaxis now than you were 20 or so years ago. Depending on how you analyze the data, that risk has actually halved. Now, I do need to say the risk is quite small in the first place. It's a very rare event, thankfully, dying from food anaphylaxis, but we're even getting better at reducing that, which is good news. But of course, that's just a tip of the iceberg. So can we get a better impression of how common food allergy is? And one of the things that we're both blessed and cursed with in the UK setting is NHS data. It's a blessing because we've got lots of it. And so, for example, the MHRA, which is our drug regulator here in the UK, runs a general practice-based data set called the CPRD, which currently covers about 7 million patients, and over the time it's existed, 19 million patients in the UK, and we're only a population of 70 million. So that's very, very significant amount of patients who are covered in that data set. The downside is that sometimes there are lots of issues in the quality of the data, how that data gets entered, what gets entered. And then the other downside is actually being able to access that with the new changes in GDPR legislation, and actually just the fact everything slowed down so much post-COVID. But we've been able to do this in collaboration with Jennifer Quint, one of my close colleagues here at Imperial and the NHLI. And so we've been drilling down on behalf of the Food Standards Agency in the UK, data on how common food allergy is. And so we're talking about incidence. This is the occurrence of new cases of food allergy, and then prevalence. What proportion of the population at any time point have food allergy diagnosis, whether it's that year or they've had it in previous years. So in terms of new cases, one can see that depending on what definition you use for food allergy, arguably, actually, it's pretty stable, maybe even beginning to decline a tiny bit in some age groups. So overall, what we did was look at food hypersensitivity, which is a bit of an all-inclusive term that includes intolerances. We looked at a sort of expanded definition of food allergy that included people who with mushroom allergy. I don't know why, but in this data set, one of the key allergens, apparently, that's really important is allergy to mushrooms. And I think I've seen one person with mushroom allergy in my life. But anyway, it's apparently really common in general practice. Then we decided to limit it to strict food allergy, where you're more likely to have a coding consistent with real IgE-mediated food allergy, which places you at risk of anaphylaxis. And then the strictest form of definition was when we said you've also been prescribed an adrenaline auto-injector device, and these are only prescribed to people at risk of anaphylaxis. So if you've got an anaphylaxis diagnostic code for food allergy and a prescription for an adrenaline pen, you're extremely likely to have anaphylaxis. And you can see that with those definitions, there's not been much of a change over the last 10 years or so. Yes, there's been an increase in the very youngest age groups, but if you then drill down in terms of, well, in that sort of preschool age group, how many are prescribed adrenaline pens? Admittedly, we might not prescribe them under 2 years of age. And then even in the 5 to 9, so primary school age groups, actually the incidence is pretty stable, which means we are still getting new cases, but we're not getting more new cases year on year. We're not in the middle of an epidemic. And so that translates over to prevalence, that yes, we are getting an increase in the proportion of people at any one time with food allergy diagnosis, but actually that increase is stable and purely due to the new cases adding in every single year as we get the next year of new diagnoses. We haven't got an epidemic on our hands here anymore, which I think is very reassuring. I'm not going to touch on whether we did have an epidemic in the past. And more importantly, because of this data set, we can get a bit of a sense of, well, how common it is. And if you look onto the government and NHS data sets, currently they might say adult food allergy is around 5% prevalence, and in kids up to 10% prevalence. Actually, those numbers are definitely an overestimate. So in adults, we're probably looking at around 1% of the UK population with a diagnosis of IgE-mediated food allergy. And that's even using quite a liberal definition. If we talk about food hypersensitivity, so very broad, including people with intolerances and irritable bowel disease and so on, it's only 2%. Now that's in many ways high, but it's not as high as sort of the 3-5% people that we see quoted currently. And in terms of children, yes, we're running at about 4 or 5% in preschool age children, and that is an increase over 10 years ago. But actually, once you get to primary age children, we're looking at 1 to 2%. So I don't want to shoot myself in the foot here. I don't want to turn around and say food allergy is not so much a problem, because it is. But I think it doesn't do us any favors to overinflate how common a problem it is. So that's, I think, really useful, valuable data that we are in the process of writing up, and is going to be informing UK policy, because it's data at a population level, not just in a smallish cohort of people living in one specific geographical area. The other thing that we also were able to look at, and we're currently doing this in a bit more detail with Adnan's team, is looking at what prompts a prescription of adrenaline, and are people religiously getting their prescriptions every year or two, or does it come in dribs and drabs, and how does age impact on that? And I think one can see that concerningly, those people with a strict food allergy, so who were very likely to be at risk of anaphylaxis, in children, only about half are prescribed adrenaline pens, and in adults, only about a quarter. And so roughly 75% of adults in the UK are not, in the last 10 years, they haven't even got a prescription for one adrenaline pen, let alone the 5 or 6 they should have had in that time period. And that's something that we're trying to look into in more detail at the moment. What about diagnostics? So this is a slide that I showed at the update last year, and it's a sort of a model of cognitive behavioral therapy applied to food allergy. And this is what clinicians are dealing with, and more importantly, people affected by food allergy are dealing with every single day. That what's driving the anxiety associated with food allergy is a perception of severity of how bad it can be, and it can kill you. We have 1 or 2 deaths per month here in the UK due to anaphylaxis and food-induced anaphylaxis. And then the perceived likelihood. And the issue we have there is that even though statistically it's incredibly rare to die, we can't predict it. So everyone, or at least parents, perceive that it's their kid who's going to be the 1 in a million, or actually the 1 in quarter of a million, who's going to die from food anaphylaxis in any year. And so it's no longer 4 per million. It's now, no, my kid is one of those 4. And so the perceived likelihood is understandably much higher than the statistics. And at the same time, people don't feel that they have an ability to cope once a reaction is happening. Is the pen going to work? Have they got the pen? Are they going to be able to inject themselves? Can they get over the needle phobia if they've got that? And so on. And what rescue factors, what things can also come in to try and make things safer? And that's what's driving anxiety. And so if we're able to reduce the perception of severity or the perceived likelihood and increase people's ability, what we call self-efficacy, to manage a reaction safely, we can reduce the anxiety. And we've previously shown that, again, using our data set, that with oral immunotherapy, where we're treating people with food allergy, 1/3 of the improvement we see in anxiety levels and quality of life is actually just due to the experience of having a safe, controlled reaction and that person fixing the reaction themselves. So there's a question there for me that's now being asked at a sort of a policy level, given the fact we can't do food challenges in everyone to try and help them address that anxiety, those anxiety drivers. Is there something we can do as an alternative? And what we know is that in terms of how people react, people don't react at the same amount. Some people react to very small amounts, milligram amounts of the allergen. In this case, these data sets are for peanut. Some people need quite a lot. So here, around 10% of people actually only react properly when they have 20 peanuts. And around 30 to 50% of people need half a peanut, again, to have any symptoms. So there's a question here to be asked, that if we can't do food challenges in everyone because it's time consuming, we haven't got the resources, can we do what's known as a low dose challenge, where we give people a very small amount to see if they're the more sensitive? Because if they're not, that might empower them to feel more confident and not to be so anxious. In my experience, every second person with food allergy thinks they're a low dose reactor, but only 5% are. So maybe we can feed people these cookies outside our clinic rooms. They're waiting to see the doctor or the nurse anyway, because 95% of them are not going to react, and the other 5% are just going to have relatively mild symptoms, bar maybe 1 in 2,500. This is something that we could be doing really routinely to improve the knowledge that people have. The other thing we can do to try and prove this is actually, is there a test? Is there a blood test that can tell us who these low dose reactors are, either so that we don't have to do the low dose challenge, or alternatively, so that we can be more selective over, okay, we're going to do a low dose challenge in you, but we're actually going to do it in a special clinic as opposed to just sitting outside the door. And so there's now a technique known as epitope mapping, which is where we specifically look at which epitopes or bits, if I'm being non-scientific, of the IgE antibody people have are able to actually bind the allergen, in this case, peanut protein. And there's certain bits of the epitope of the IgE antibody binding region that seem to not only predict a little bit in terms of how people react, but also the amount of allergen they'll react to. And so there are now a number of publications that our Imperial cohorts have contributed to in terms of developing this test, which I think is going to be a significant improvement in terms of giving people more specific personalized advice, rather than treating everyone like they're all at risk of having a really bad reaction to a tiny amount of the allergen they're allergic to. And so you can see these data here, where by this epitope mapping, looking at 64 different binding regions, there are 2 or 3 regions that together are able to predict with pretty good certainty, 95% certainty, if you're a very low dose reactor or a medium dose reactor or a high dose reactor. What about immunotherapy? So we're slowly getting round to desensitization, beginning to have this enter routine clinical practice, although it's really slow because there's no real funding in our health system for this, for the actual clinical visits. And we've basically got 3 modalities available. We've got oral immunotherapy, where you have the allergen by mouth every single day. We can stick the allergen underneath the tongue, and we can stick it in a patch. And I'm going to sort of focus on these latter 2 at the moment. There's a lot of interest here with oral immunotherapy. We've got a peanut product available on the NHS, but uptake's been very slow at the moment because of the resourcing issues that we have. This is the EPIT. This is a phase 3 study that's currently going through and about to be published in New England Journal of Medicine. It's a study that we contributed to. Imperial was the lead UK site, where we demonstrated that in young children, so this is kids aged 1 to 4 years old, sticking this plaster on the skin every single day was able to induce a significant degree of desensitization. It is much more modest than we can achieve through oral immunotherapy, but it is associated with far fewer side effects. And also, you don't need to come every 2 weeks to hospital to go up on a dose, but you just stick a plaster on and that's it. You do it every day at home, job done. That may offer more considerable improvements in cost effectiveness than oral immunotherapy, although we need those comparisons to be done. So that's quite exciting. But the other thing that's very exciting from the Imperial team is the SOCMA study. And this was a protocol that we followed that actually Marta was very involved in as well, where we wanted to take people with more difficult milk allergy. And I'm delighted that Pablo Rodriguez from Madrid is on this call as well. I saw him there, and he was also the co-investigator, because there's a 2-center site between London and Niño Jesús Hospital in Madrid. And what we did was say, rather than stick people straight on oral immunotherapy, can we be clever here? Are we missing a trick? Can we do pre-treatment? And by pre-treating them, we can make oral immunotherapy more effective and safer. And so some people were randomized to placebo pre-treatment, that is nothing. Some people got sublingual immunotherapy with milk, and some people had the same dose of sublingual immunotherapy, but they swallowed it straight away. They didn't stick it underneath their tongue. And so that way we could see, is pre-treatment doing anything? And on top of that, is there anything special about the sticking it underneath your tongue route? And so they did that for the first 6 months. And then the second half of the study, everyone had conventional immunotherapy. And as you can see, these are the screening the demographics of our group who are screened. And the key issue here is in terms of how much milk people reacted to at the start. And you can see that the randomization fortunately worked. And we had a nice spread, normal distribution of people of similar levels of sensitivity across the 3 treatment groups for the first half of the study. So this is what happens. As you can imagine, and it's not unexpected, those getting placebo, not a lot happened in the first 6 months. And then in the second 6 months, when they were on actual treatment, they got desensitized, the amount that they tolerated, the top amount of milk they tolerated, and here we're talking around 60 to 100 mils of milk, that went up from what we're talking about here, which is 5 mils or even less. In those who had pre-treatment, one can see that there was also an increase, and that increase was significant over the first 6 months. And then the second 6 months, it was more modest, and they all got to the same place. So arguably, eventually you'll get to the same point, how you do it. The question is this safer than just doing it in 6 months with the conventional route? And so that's data that we're looking at the moment. And we are quite excited because the initial data shows that for the first 6 months, there was more of an increase in those people having the sublingual route than in the people having the same dose by the mouth not being kept underneath the tongue. And so arguably, these are the first data ever that the sublingual route is not just taking being exposed to allergen by mouth. We use sublingual immunotherapy for hay fever and for dust mite allergy and so on. But the reason there is because you can't swallow dust. Well, we are swallowing dust mite, but we're not going to talk about it on this call because it's a bit off-putting if you're about to eat your dinner. But there's not much evidence that, we don't really know whether the sublingual route is special or whether actually it's just because it's being exposed to the oral mucosa and then the sort of upper gut. And these are the first data, because it was a study done in food allergy, that shows maybe there is something special about the sublingual immunotherapy. It's going to be exciting to see if we see that difference reflected in the blood tests and various other investigations that we've been doing. So then finally, what about severe allergy? So this is a study that we're very excited about, that we're currently trying to get set up. We've had funding awarded, and this is the IRIS study. And what we're doing here is identifying people who've had near fatal anaphylaxis in the UK to food, but also other triggers. And then we're going to look at them and see if those people are different from people who have just bog standard anaphylaxis to the same triggers, and the main one being peanuts in this study. And so we're really going to be drilling down to identify, is it just bad luck when people have near fatal reactions? Or actually, is there something in their mast cells, their basophils, the effector cells that cause the allergic reaction, or something in the circumstances? Is their asthma not as well controlled? Is there some genetics influence or something like that that can explain why some people react so badly and most people don't? Because if we can identify some risk factors, we're really going to be able to move towards personalized medicine for food allergy. So I'm going to stop there, and I think we've got time for questions. So I'm going to hand back to Marta.
Lovely, Paul. Thanks so much. I have to say that was really the latest, because I hadn't seen the results from the SOCMA study yet. So I was very happy to see this graph, as you know, Paul. Lovely. Great. So we have a question in the chat. It's about, sorry, it's just disappeared right now. Just a sec. The question is about, just before I'm not managing to get it on, but anyway, is about whether, you've shown this discrepancy between the prevalence you're getting with the study you've done and previous publications. So someone was asking, are we going to start seeing these kind of lower figures quoted in places like the NHS website, etc.? So I guess the implicit question there is like, how does, say, the quality of the data you're producing compared to other studies that have been done before, and what sort of figure do we go with?
So I think the interesting thing is that we have had some really good challenge studies done in the UK. We've had the Isle of Wight suite of studies that were led by Southampton, Karina Venter and Hassan Ashad in Southampton. Not everyone got challenged. Some people said no, but it's not bad for a study in a decent geographical cohort. And then we've also got some data from the control arm in the EAT study that Michael Perkins ran, looking at earlier introduction, because it wasn't, unlike the LEAP study, it was a more normal risk group of people in the UK population. The only quirk is that everyone was exclusively breastfed in entry, which makes it very different from what happens normally in the UK. But figures in terms of peanut allergy and egg allergy in both those studies actually match what we found using the general practice data set, around that 1 to 2% mark by the time you get to primary school age. Now, egg allergy is more common in babies age 1 to 2 years. But certainly for peanut allergy, it's quite concordant. So that's reassuring that even though the general practice data isn't based on food challenges, I'm not saying we don't need food challenges for epidemiological data, we do. But the fact that the percentages in those preschool, so those primary school age children are pretty similar to the studies that have used food challenges, to my mind, acts like, you know, one data set is almost validating the other and vice versa. So where we start seeing these figures of 5% of kids with food allergy, maybe they are at age 1 to 2 years, but certainly by the time we get to school age, we've got food challenge epi data, and now we've got general practice data going, actually, it's more like 1 to 2%. And so actually, it's that agreement that I think is really key. So where it's interesting in terms of adult allergy, and there is a study funded by the FSA at the moment, the PAFA study, that is going to involve food challenges in cohorts of adults. So it'll be interesting to see whether the data coming from that study matches what we found in the general practice database.
And do you think resolution of food allergy in young children is well captured in that sort of data you're collecting in terms of, you know, prevalence over time?
So it would be in incidence, because incidence is only new cases. We have looked at it in prevalence, and that's one of the reasons why we wanted to use a strict definition that excluded a old diagnosis of egg allergy, because we didn't want, you know, we were worried that if you diagnosed with egg, and often egg is one of the allergens that people outgrow, you'd be labeled for life under CPRD. So we did look at some of these issues to try and sort that out. And obviously, if you have got resolved food allergy, you're no longer going to be prescribed an adrenaline pen. And that's why, for me, that's the best definition that we've used to analyze these data.
That's great, Paul. And there's a question from Kate Swan asking what the age range is for the IRIS recruitment.
So at the moment, for the IRIS study, we're recruiting adults, but we're doing it through a clause in data protection legislation that involves us being able to identify people through NHS data sets. So we're not asking people to contact us or to refer patients into us. We're actually going to be writing in due course, we hope, to everyone in the UK who's ever been admitted to intensive care to say, are you willing to take part in this study? And that's the real strength here, because we're going to have a national unselected cohort. So we're going to be minimizing the selection bias that has plagued previous attempts. It's much smaller attempts to do this. It's the first time this sort of case control study has been done at a national level anywhere in the world. And we can only do that because the way the NHS data is set up.
That's fantastic, Paul. I don't think there are more questions in the chat. I have a question about epitopes. Really, really exciting what you were presenting. I know there's been a previous research looking at epitopes for peanut, looking at success of peanut immunotherapy, safety, etc. And I believe severity. Has anybody compared whether, say, the same epitopes, the same regions, kind of not only might predict thresholds, but also severity?
So we are looking at that in a number of our studies at the moment. We're looking at that in terms of the peanut studies, but also SOCMA. And the really exciting thing in SOCMA is that we've got two different techniques. So we've got one research group that we're collaborating with closely in Madrid, and then another research group in the USA, where we're going to be using different techniques to look at the changes in epitope mapping and IgE binding prior to and after immunotherapy, to see if this can give us a bit of a clue in terms of people who are more likely to have straightforward treatment courses or, you know, bump around a lot, or alternatively to see whether we can use that to select people who are going to respond better to immunotherapy, because we don't really have good predictors at the moment, as you mentioned.
Fantastic, Paul. Thanks so much for joining today. I'm sure people have enjoyed your talk and the Q&A. Thanks so much, Paul.
Thank you.
Lovely. We're going to move on to Adnan. So Professor Adnan Kustovich, there's a little introduction for a very outstanding CV there. So Adnan is the lead of the Pediatric Allergy Group within the National Heart and Lung Institute and the Centre for Pediatrics and Child Health at Imperial College. His research over the years has focused upon the origins and natural history of asthma and allergy across the life course, with an emphasis on prevention and translation of patients' benefit. He pioneered the use of data-driven methodologies in the analysis of complex data, including the first use of machine learning in respiratory medicine and allergy. He's been instrumental in a number of birth cohorts and consortia over the years, including Stellar and now currently the MRC Unicorn program, integrating birth cohorts, patients cohorts, and randomized controlled trials for joint analysis, trying to understand the mechanisms underlying different asthma endotypes. Adnan has a number of awards from different, renowned institutions, such as the European Respiratory Society Gold Medal in Asthma, and he's extremely active in research and in academia. So brilliant to have you today, Adnan, and over to you.
Thank you so much, Marta. Thank you for that really kind introduction. And obviously, Marta has asked me to talk about disease trajectories, but rather than focusing just on data-driven stuff, machine learning and AI, I decided to look at it from a slightly different angle. The beauty, as I see it, of Imperial is this unique ability to rely on three important pillars for the research. Everything obviously underpinned by clinical research, but clinical research is not enough. You really need to have wet lab, basic science to explore mechanisms, and dry lab, data science, to really make sense out of increasing quantity of data that we're having. So I'm going to tell you a little story here, that is about wheeze phenotypes. So decided not to touch upon food allergies or anything else. And most people kind of relate wheeze phenotypes to Fernando Martnez, but the story of wheeze phenotypes started earlier. And it's really sort of this phenomenal observation, clinical observation by Nicola Wilson, captured in extraordinary rostrum article published in '89. And I urge everybody to read it because it is so good. Such an ability to apply a thought process to clinical observations. And it was a day and time when people were trying to persuade everybody to start with anti-inflammatory treatments for pretty much every patient who wheezes. And what she argued was that wheezing illness, particularly in early preschool years, is highly heterogeneous. And she said then that the description has always been confused. So quite frankly, nothing really has changed over the period of last 25 years. And she continues to explore this idea of different types of responsiveness, viral responsiveness, atopic responsiveness, and bronchial responsiveness that then overlap and produce different clinical phenotypes. She argued that what she sees in clinical practice is that some children start with wheezing early and then stop, never to wheeze again. Others continue to wheeze into their school years and then into adulthood. And there are some kids who don't wheeze early, but wheeze later on. And this whole idea of different temporal patterns has been born. Fernando Martnez was inspired by this, and he tried to test this idea of different temporality in Tucson Children's Respiratory Study, and lo and behold, described these famous three wheeze phenotypes: transient wheezers, starting early and stopping in his hands by age 3 years, late onset wheezers starting after 3, and persistent wheezers kind of going throughout preschool years. Now, why birth 3 and 6? Is there anything special about these time points? Well, no, it's just this is when data was collected in Tucson study. So Fernando really tested this clinical observation in a context of clinical research study to describe different wheeze phenotypes. And the term phenotype stuck. What does it mean, phenotype? Well, phenotype is really what you see. And there lies a problem, because the problem with what you see is very often you see what you want to see, not what is necessarily there. So when I look at this presentation, I look at my sort of the camera, I see the young guy with blonde hair, which is complete and utter nonsense. And we always ask the question, well, actually, rather than trying to force our hypothesis on the data, how about trying to explore the patterns within the data sets to see what actually is hidden there? What is the hidden structure, the latent structure? And that's where machine learning is particularly good. It really kind of identifies patterns within large data sets. Now, whether these patterns are meaningful, whether these patterns are related to pathophysiology, is a different question altogether. So most of the data that I'm going to show you today is from our birth cohort consortium, Stellar Unicorn, comprising 5 birth cohorts, 16,000 individuals followed from before birth, 8 weeks of pregnancy, now up to the age between 24 and 34. So we have a life course from before birth into early adulthood, trying to capture the patterns of disease. And the first paper from Stellar Consortium was the famous analysis by John Henderson and Raquel Granell, when we applied what is a data-driven methodology called latent class analysis to try to understand what is the hidden pattern. And what the data told us was there appeared to have been not 3, but 5 different phenotypes of wheezing, persistent clearly there, late onset clearly there, but then transient wheezers could really stop very early, later on, and there were a group of kids with intermittent wheezing. This was done only in the context of ALSPAC and up to age 6 to 7 years. Now, once we managed to put all of our data together into the sort of knowledge management platform, harmonized and integrated, we were in a position to repeat this analysis, but now on longitudinal data from birth into early adulthood in all of our study participants, all 16,000 of them. And the structure in this mother of all latent class analysis suggested that there are indeed different temporal patterns. There clearly is persistent wheezing, late onset wheezing, and in terms of transient wheezing, 2 different patterns, early transient and those who kind of stop wheezing somewhat later. Now, this is kind of really big data-driven analysis about patterns. But are these patterns really different diseases? Are they consequence of different mechanisms? And it is a very important question, because if they are, then obviously the way you treat them, the way you approach patients should be fundamentally different. But how can you make inference, try to reach conclusion out of the clinical data only? Well, you can't. And the question that we asked was, okay, now that we have this structure, how about asking question whether different wheeze phenotypes are underpinned by different genetic associates? So if we run genome-wide association study, not trying to ascertain associates of childhood asthma, but different wheeze phenotypes, what will the data tell us? And Raquel Granell then did the first example of genome-wide association study using multivariable outcome. What do I mean? So here we are comparing children without wheezing to those in 4 different phenotypes of wheeze. Most of you will be familiar with standard Manhattan plots, where you have, association with, let's say, asthma. This is circular plot. What does it mean? So every little circle here represents different phenotype of wheeze. Persistent wheeze, transient preschool resolving, transient mid-school resolving, late onset wheezing, trying to capture the genetic architecture of these different wheeze phenotypes and asking the questions whether they differ. And indeed they do, because there are unique associates of each of wheeze phenotypes. 44 SNPs uniquely associated with persistent, 25 SNPs only with preschool remitting, 33 with mid-school remitting, and 32 with late onset wheeze. So completely different structure. And interestingly, this association analysis, this genome-wide association analysis identified a hit in chromosome 9 close to an exon 1 gene, a genome-wide significance 10 to minus 9 that was uniquely associated with persistent wheeze. Okay, then you need to think about functionality. So we looked at promoter capture high C interactions to demonstrate that the SNP, which we identified here, which was associated with persistent wheezing, overlapped a region which interacts with promoted an exon 1 gene. Now that suggests that this is likely causal variant. In EQTL analysis, this genetic variant that is associated with persistent wheeze is also associated with expression of protein. So it makes more sense. And the biological function of an exon 1 is really interesting because it interacts with genes that are enriched for asthma and other T2 diseases, including IL-4 and IL-13, and also glucocorticoid receptors. So suddenly it makes a lot of sense. Now, the golden rule in genetics is replication. Now, you can replicate or try to replicate analysis in which you're looking at associates of asthma. But this type of analysis is impossible to replicate because it's unique data-driven. And to replicate it, you would have to have precisely the same data points in various cohorts, which simply does not exist. So you're kind of stuck. You have really interesting things that make sense. What can you do next? And what we can do next is probe the functionality. That's where the power of wet lab comes in. And our colleague Claire Lloyd, who is also teaching at this course, a fantastic then series of studies looking at functionality in house dust mite model, in mouse model, looking at the expression of an exon 1 in mouse model of house dust mite induce allergic airway disease. So it is adult model, obviously, because we're looking at persistent wheezing. And it's staggering because in immunohistochemistry, an exon 1 protein clearly is increased following house dust mite exposure, and mRNA is clearly significantly induced in lung tissue after dust mite exposure. And you can then argue that perhaps pro-resolving an exon 1 may play a role in the way we regulate lung responses to an allergen, and therefore it can really be associated with persistence of wheezing. Now, they also created a knockout model of mice deficient in an exon 1. And again, consistent with this idea, mice deficient in exon 1 had markedly worse allergen-induced lung function, greater airway resistance. So it's really sort of expressed in this physiological outcome. And also, they appear to have significantly more eosinophils and T2 lymphocytes. And finally, lung tissue cytokine levels reflected the exacerbated T2 inflammation with elevation of IL-4, IL-5, and IL-13. So you see how you get from clinical observation in 1988 through data-driven methodologies which have been developed and applied over a period of 20 years to then wet lab to try to probe mechanisms to come up with a brand new therapeutic target. Now, admittedly, this is fairly rare genetic variant, and we can explain only small proportion of persistence of wheezing. But if we can come up with proper target, I mean, that would be great. So, you know, that all looks really great. But when you look at some detail at those famous wheeze phenotypes, everybody tells you different studies come up with very consistent structure with same wheeze phenotypes. Well, the truth is, the phenotypes or the latent classes have the same name, but they don't really look that similar as you would want them. So wheeze phenotypes discovered in different studies have the same name, but their characteristics differ. So transient wheeze can end up anytime between 1 and 10 years of age. Late onset wheeze starts between 3 and 11. They're not the same. And we've done a number of studies trying to ascertain precisely what's going on to demonstrate that sample size, frequency, timing of data collection has major influence of the structure that you have within the data point. For those of you who want to do research, these are the key time points. And the final problem is, in all of these models, a lot of kids may be classified imprecisely. So we did a hell lot of analysis where we excluded one cohort, did incomplete data set. Whatever we do, the population structure is the same. So you think this is great. Well, population structure is the same, but you in a clinic will not see population, you will see individual patients. So this is what we show you in papers. It looks really lovely, lovely, lovely graph of different wheeze phenotypes and patterns. If you ask questions, well, are these internally homogeneous and stable? This is what you want if you are going to do large genetic studies. What do I mean internally homogeneous? Well, do children who cluster in these different phenotypes actually look the same? What does it mean stable? If I apply model to, let's say, those with full data set and those with imputed data, will the same children always end up in the same cluster? So that's the graph we show you. This is the graph that you would really like to see. So this is, about 15,000 individual children. The individual patterns of wheezing across childhood. And what you see is that quite a few of children who never wheeze actually have reported wheezing. That late onset wheezing can start at any time point between 3 and 14 years. That there are quite a number of transient wheezers who wheeze when they're 18. I mean, some transients, that is. And there's a lot of heterogeneity in terms of persistent wheezing. So they're not internally homogeneous. Why on earth does this child here with identical pattern of wheezing to this here end up in 2 different classes? Well, it's just the consequence of methodology that you use. Are they stable? So what we did is we took kids with full data set, about 7,500, and then ran the model in those with imputed data set, 16,000, asking the question, do children with full data set end up in a same class as they do when we run the model with imputed data set? And the problem is there's quite a lot of transition. There are 2 very stable phenotypes, persistent and never. So all of the data that I've shown you about annexin-1 holds true. What I want to know clinically is, well, actually, if I have a wheezer in front of me, can I predict whether the wheezing is going to continue or not? And this is not the optimal model, because what you can see is 25% of children in these intermediate phenotype switch the phenotype. So the question is, can we do better? Can we come up with better and more stable and more internally homogeneous phenotypes? One of the postdocs in our dry lab, Sadia Hader, who comes from really sort of slightly different background of economics and then also social sciences, who did a lot of research into poverty, said, well, you know what? You're asking a question of persistence of wheezing based on a simple presence or absence of wheezing at discrete points. But when you look at poverty, for example, it's not whether you have been poor or not. It's particularly at which point in your life course did you fall into poverty? How long did it last? Did you recover? What was the pattern? She said, I want to do the analysis where I take this much more holistic view of wheezing. And she did. She developed 6 multidimensional variables of wheezing spells. So how long did they last? When did they start? When did they stop? What was the pattern of persistence and recurrence? And then applied different type of clustering to identify slightly different structure. Indeed, you have persistent wheeze. Now look at this. Late onset wheeze, they all start at about age 8 to 10. Transient wheezers stop wheezing by age 4. And there are two persistent groups. One genuinely persistent, and one which we kind of described as intermittent, which makes sense for all of you who have sit in a clinic. Some children who have symptoms all of the time, and others who just crash and burn from time to time. So the question is, are these now internally homogeneous? Well, indeed they are. Not a single never wheezer have ever wheezed. Late onset wheezers, they don't wheeze in early life. They start wheezing 8, 10, 11 years. Makes all the sense in the world. These are your mild allergic asthmatics. Most, if not all, transient wheezers end up wheezing by school age, and there are two different patterns of persistence. Is this now also stable as well as internally homogeneous? Well, yes, whatever we do with the data, however we extrapolate it, same children always end up in the same cluster. This is really now exciting because we have very stable and very internally consistent phenotypes. And the next step, obviously, that is currently going on with our colleagues from around the world is replication, genome-wide association studies, and mechanistic studies. So some of you who may be interested in master's projects, please do come. There's a lot of stuff to be done here. So what is the structure of this? Well, clearly they differ in their genetics because the famous 70q21, childhood asthma locus is associated with persisting but not transient and late onset phenotypes. Makes absolute sense. What is childhood asthma? Well, childhood asthma is a mishmash of all of them. 6% of kids who have, or adolescents who have diagnosis of asthma have never wheezed. Quarter of them are intermittent. Third of them are persistent. Quarter of them are late onset. And actually about 6% of them who have asthma diagnosis in adolescents have never wheezed since the age of 4 to 5 years. So it's really interesting that even amongst persistent wheezers, kids who have consistently wheezed, third of them do not have doctor diagnosis of asthma. Now, some of my colleagues tell me, well, that's because their doctors don't have a clue of how to diagnose asthma. But personally, I don't believe that is the case. Their GPs, their primary care physicians, sees the kids all the time. They observe patterns. They know more about them than we do. This is also transferable. So we very recently applied it to a birth cohort in South Africa. Again, remarkably stable and internally consistent structure, which although you have transient late onset and recurrent wheezing, it looks different than what we see in the UK. And the associates are very different. In African population, it is not rhinovirus, but RSV, lower respiratory tract infection, that is associated with increased risk of recurrent wheezing. So the pattern does not necessarily tell you about the mechanism. And it's really exciting because here we have also lung function data that demonstrate very clearly that transient wheezers start with impaired lung function at age 6 weeks, which then somewhat improves. Persistent wheezers and recurrent wheezers have impaired lung function, and then it further kind of deteriorates over the first 6 years of life. So there is a window of opportunity, and we can do something about it. So thus far, what you can see is we can really utilize team science to understand this. Wheeze phenotypes, now this recent development where we use spells, appear to be clinically more intuitive. There's great internal consistency and homogeneity, much more robustness to imputation. Does this mean that they are more reflective of mechanisms? Well, hopefully we'll tell you next year or in a few years' time. This is really ongoing work. But 5% of those with doctor diagnosed asthma have never wheezed. They have something. 30% of those with persistent wheeze don't have asthma diagnosis. So can we come up with more clinically intuitive subgroups which reflect mechanisms better by applying these complex data analysis techniques to the information on all of the symptoms? again, one of the things that is being asked by one of our PhD students, Alex Cook on. And in the end, you know, a hell lot of people, not just Imperial people, have contributed that. Angela and Claire from Manchester, Raquel from Bristol, Steve Turner from Aberdeen. Sadia has been instrumental, as well as Paul here at Imperial. In terms of our colleagues, is John Graham and Hassan from Southampton. Extraordinary data science team. This is clinical research outfit, extraordinary data science team, Sadia Hader and Sarah Fontanella, all of our friends and colleagues who are exploring the mechanisms, particularly Claire Lloyd with mouse model, again, critically important, and friends and colleagues from around the world who have contributed to this in terms of replication. Any questions you may have, happy to answer.
Thanks so much, Adnan. That was absolutely brilliant and lovely new data there as well. I don't think we have any questions in the chat for now. You focus very much on asthma and wheeze. Would you like to give us, you know, just a snapshot on data about the other allergic diseases and sort of the concept of the atopic marsh? And I'm thinking about the paper from Sadia as well from last year with these very nice graphs showing, you know, atopic eczema and progression of different diseases.
You know, I mean, the whole atopic march concept is so attractive and people fall in love with it because of this faint idea that they can do something about the progression. Now, you know, march means something. March means moving in unison, and atopic march means you're moving from eczema into wheezing, asthma, into rhinitis. And that's obviously a complete fairy tale and fantasy, because that's not what is going on. Really, what is important to understand is that there is tremendously important phenotype of atopic or allergic multimorbidity. These are the kids with eczema, with wheezing and asthma, with rhinitis, food allergy, because obviously, you know, the only birth cohort, insofar as I know, or the first birth cohort to have objective data on oral food challenge confirmed food allergy is our cohort in Manchester. And the data there were bang on what Paul is talking about, you know, prevalence of peanut allergy confirmed on oral food challenge is 2%, thanks very much in school age. Now I think Angela and Hassan are doing the follow-up of Isle of Wight and Manchester to look at what happens in adulthood. So there is tremendous variability in the way individual children develop symptoms, but what is very important for all of you in practice, children with allergic multimorbidity declare themselves very early. By the age of 4 or 5 years, you have the whole lot. After that, it remains remarkably stable all the way to age 30 years. There is no march. There is allergic multimorbidity, and we need to focus on this and understanding the mechanisms, okay? Most two disease kind of combinations are just a pure chance. So, you know, in essence, you have children who have mainly asthma, mainly eczema, mainly rhinitis, and those with multimorbidity. What we need to understand is what are the mechanisms of multimorbidity. Out of 10 children with eczema, only 2 go on to develop allergic multimorbidity, okay? 2 have resolution of their disease by the age of 30, and 6 remain having eczema alone. So the idea that eczema just disappears is wishful thinking. The idea that children, individual children with eczema had higher risk of having asthma, well, that is true at population level, but surely not true for that individual child whom you see in the clinic.
Lovely, Anna, thanks so much. I'm just saying that I believe there is a question in the chat just coming in, so I'm just reading out loud, so we will finish after that. So Walid, one of our students, very interesting data. He's saying, until we know more, what do you foresee the implications for therapy are, specifically for acute asthma or wheeze?
We actually know more. What I haven't shown here is a very detailed investigations that we did with Sadia in terms of pathophysiology underpinning preschool wheeze. Okay, so, and this is done in severe preschool wheezers in whom Sadia has done bronchial biopsies. So they present with very, 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 very similar, if not identical, clinical presentations, severe episodes of wheezing that actually really sort of was sufficiently severe for Sejal to consider bronchoscopy. More than 100 of these kids. When we apply these complex analytical techniques, it was very clear that there are 4 distinct pathophysiological clusters. Okay, 25% of preschool wheezers are characterized by eosinophilia and sensitization. That's 1 in 4. 25 or thereabout have very clear signs of infection, both bacterial and viral, in their airways. So it's predominantly infection driven. 25% appear to have reversible airway obstruction. 20% of them look perfectly normal. If you think about all of the data that we have, and for example, data from infant study, it was very clear in the suggestion that only preschool wheezers with atopy and peripheral eosinophilia respond to inhaled steroids. So only 1 in 4 preschool wheezers will have benefit from inhaled steroids, and yet in our hands, 90% of those who come to us have it prescribed. So this is nonsense. In 21st century, prescribing data for preschool wheezing based only on clinical observation is not good enough. There's, insofar as we're concerned, and our proposal is you need to do a set of investigations. Full blood count for peripheral eosinophilia, you don't need to do it. You can pick it up from healthcare records. Skin prick tests to common inhalant allergens. If both of these or one of these is positive, by all means prescribe steroid. That will be good for 1 in 4. Please make sure that you confirm wheezing. Confirm also, if you can, using FOT or using your stethoscope, confirm reversibility, wheezing before and after administration of salbutamol. You can use very simple NPA to determine infection. So those with peripheral blood eosinophilia and sensitization should be prescribed steroids. Those without these 2 features, with confirmed low airway infection, let's say on NPA, give them antibiotics based on specific findings that you have. Those with neither of the 2, where you demonstrate very clearly airway obstruction, go for bronchodilator only, or ipratropium. I have great hopes that tiotropium may also be here. So there's stratification of treatment that is based on objective markers of what are treatable traits in this age group. People give steroids. They are not smarties in this age group. Giving inhaled steroids to infant, you need to think about that very, very carefully. Fantastic drug for those who are sensitized and have peripheral eosinophils.
Great, Adnan. Thanks so much for your insights. Thanks so much, Adnan and Paul, for joining us today for these brilliant talks. And thanks to all the attendees for being there and the interesting questions there. So lovely. I wish you all a good afternoon. And if you're interested in any of the programs, please get in touch. There are a couple of useful links in the chat. So again, thanks so much for attending. Bye.